So I will talk a bit about genetic and genomic methods. Again, it will be short, but give a bit of an overview of how we can use this for analysis of populations and for hybrid detection. So in short, what can genetic and genomics offer for conservation? We have been using these tools to examine species, population distribution. We can look at extent of gene flow, and we can identify key areas for connectivity, which is really important for conservation planning. We can also quantify genetic diversity and inbreeding, which helps prepare conservation strategies. Also, there are some different and interesting processes now going on the landscape that we need to be aware of and where genetics can help us. For first thing, we have rewilding and recolonization. Many species are expanding their range, and we are seeing increased contact among long isolated populations. And generally, this is seen as beneficial and positive for conservation. However, now when we have migrant individuals, we need to be sure we are not mixing up these with animals that are results of domestic wild hybridization. This is also a concern for many species, terrestrial and aquatic, and something that occurs across the globe and across taxonomic range. So I will briefly talk about three approaches. First, continue a bit on an example on microsatellite markers that Tomáš presented, then talk about the use of genome-wide SNP panels, and finish with a presentation also a little bit about how reduced SNP panels of discriminant markers can be an important help for analysis of hybridization. So if we look at microsatellite markers, we are using these now for a long time to look at population structure and genetic diversity. As Tomáš mentioned, there are more traditional or standard procedures and also these newer high throughput sequencing. And I will briefly talk about an example where we use the traditional method. And these markers are highly diverse. Each has many different alleles. They're therefore excellent for individual identification. And this is very important for many populations of conservation concern, because we may then have low genetic diversity and high genetic similarity among profiles of individuals. So my example is a study we did on the Mediterranean monk seal. This species is mostly found in the Eastern Mediterranean, and it has a vulnerable status with only a few hundred individuals, according to IUCN. They have a unique breeding behavior where females are pupping in marine caves. And they are a marine conservation flagship species because their interactions include many of these wildlife-human issues that concern, for example, pollution, getting entangled in fishing nets, overfishing, etc. And we examined 86 animals that were found dead around in Greece over a decade and used DNA from tissues and analyzed these with 26 microsatellite markers. And when we did this, we found some signs of population isolation and low genetic diversity and high inbreeding. If you look at the plots I show here, you see a result from analysis of the program structure, and each vertical bar is one individual and the ancestry of that individual. And you will see that on the right side, there are some animals in green that are really distinct. And this is the isolated population in the Ionian Sea that if you look on the map on the left side, you see this showing also geographically as an isolated population, although we do see one migrant animal in red. So these type of markers are very suitable for non-invasive or other analysis where we have samples of limited quality, this often happens in species of conservation concern. We don't want to kill them or disturb them to do our study, but here we could do a study on the decomposing tissue of these found dead seals. So this is a very good way because we can use such samples for our research. And they are hypervariable markers. Again, they can identify individuals with relatively few loci. And they are fairly common in the sense that there are lab setup, the analytical experience is fairly well established. This also means that because it is an established method, there are often long-term legacy data sets. People have been collecting data for many years, and we can therefore more easily detect population trends, which is also crucial for conservation work. In terms of pitfalls, Tomáš already mentioned that these have genotyping errors, and we therefore need to analyze each sample multiple times. Some of them may not work. We have also that the traditional microsatellites need standardization among labs. So this is costly and also time-consuming. And of course, if we have these problems, this is tricky for transboundary issues and of studies of many wide-ranging species when you want results for a wide area and when you need quick results for rapid conservation decision-making. Then if we move to genome-wide SNP panels, these are, as microsatellites, biparental in inheritance, so you get information on both the mother and the father's side. They are a point mutation, so a change of one letter in the genetic code, for example, an A here in green substituted for a G. And this use of the very basic genetic information means that they are compatible between labs and we can put our data together. And they're also used to study both neutral and adaptive processes, for example, where we're interested in selection on specific traits. These panels were first developed for species of more commercial interest, for example, cows or pigs or dogs. But that means that they are also very useful for the related wild species. So for cow, pig, and dog panels, we can also use them for bison, wild boar, and wolves, as we have been doing. And very interestingly and positively, they're also increasingly available for non-model species. And one example of this is a very nice study done by our colleagues at University of Ljubljana, where Recknagel and colleagues recently published a very nice study where they looked at the Proteus cave salamanders with over 40,000 SNP markers. And in terms of application, here we use DNA extracted from tissue and blood to get enough quality material for these genome-wide panels. So we are limited to more of these high quality materials. And we do need data filtering and analysis that use more powerful tools than the often Excel or simple tools that you can use for microsatellite analysis if you have only a few markers. But we also get data sets of usual large quantities, such as 10,000 or maybe 100,000 of markers. An example I want to show is a study we did on European wolves, and here we did combine data developed by different labs. We used the canine SNP panel with 170,000 SNPs that was developed for domestic dogs and analyzed 177 samples across Europe. And we found 5 major clusters as shown here on this map. We could also then go back and look at some results that have been made with a smaller number of microsat markers. So we could confirm divergence observed across relatively short distance when we know how far wolves disperse, seen with microsats. For example, if you look at the map, you can see Carpathian wolves are distinct from wolves in northern Poland, although this is not a very long distance for wolves. We also saw north-south structuring in Europe, and we could, again, go back to older microsat results and see that we can confirm with these genome-wide profiles that Italian wolves are indeed the most isolated and most distinct. Another example is a study of wild boar that I did with colleagues in a study led by Jakolina et al. Here we used the porcine SNP panel with over 50,000 SNPs developed from domestic pigs. And we analyzed a large number of animals, wild boar and domestic pig breeds. And we found at least 33 wild boar that had clear signs of domestic ancestry. These are mostly found in Southern Europe, and probably is a question of whether people have free-ranging or industrial farming. And these more free-ranging cases where there is contact between animals, this clearly affects the ability for hybridization, but it also has very large implications for African swine fever for conservation. In here, I just want to draw your attention to the map, and there are lots of colors here which are looking at different kinds of populations. But if you look at the markers, you will see that the round markers are wild boar, and the triangle markers are domestic pigs. And we can see that there is clear population structure and also some signature here in the middle of likely hybrids that seem to have been misassigned, like this triangle here among the circular ones. And you do see that this structuring of all these populations still is a bit of a noise when we want to look at hybridization. So what we did then was identify about 1,000 ancestry informative markers. So we analyzed the same animals again, just with those fewer and small panel with 1,000 markers. And you can see that this really improves resolution for hybridization. So advantages of using the SNP panels are that these usually give you large data sets. This means also this method that it allows you increased data compatibility across labs, which is greatly beneficial. And we can do both in-depth and wide-scale analysis of population structuring. We can do identification of discriminant markers, like we showed here for the wild boar. And also, we have powerful, freely available tool like R packages for data analysis. And I think this is worth mentioning because many conservation workers, they may not have a very huge budget, but if they can use data that is freely, or data and tools that are freely available, this really improves the ability to do such analysis. There are also some pitfalls to mention because we need high quality DNA. This means that such analyses are usually limited to invasive sources such as tissue and blood. And these large data sets have many advantages, but there are also the issue that you then need more tools, computer power, and analytical training than you would do for a smaller number of microsatellite markers. And importantly, panels made for one species may have a bias when used for other species. Wolves and dogs, for example, are closely related and it works well, but I've also worked on analyzing such data for coyotes, which you can do on this chip, but you will get some biases and they seem less genetically diverse than they actually are, just because markers were actually selected for dogs. And then for the final topic of the reduced SNP panels, we see from the wild boar example that discriminant markers for hybrid detection can be found within these large SNP panels. But very importantly, even if we only want these SNPs, that means we still have to have good enough DNA to use these methods. So that makes, that gives us a challenge. But however, once these discriminant SNPs are identified, these reduced set of markers can also be developed for uses that require less DNA and is more friendly to non-invasive samples, such as the fluidigm method that Tomáš mentioned earlier. And an example I want to give here is a canid hybrid SNP panel. This is part of a study that we participated in and was led by a colleague from Finland, where it was developed as a panel of 96 discriminant SNP markers from this panel. So we could then use these to identify wild versus domestic canids across Europe. and identify not just the first generation hybrid, the F1, we're sharing this picture, but also the first level of backcross. So if this hybrid crosses back to a parental species, for example, a wolf, and the second level of backcross. And just to show this as a figure, on this plot here, you can see in green the domestic dogs, and you can see on the other side, on the left side, various wolves. And here you will notice that I mentioned Italian wolves being very isolated. They are shown in yellow, and wolves from other areas like Northern Europe, Finland, Russia are shown in blue. But here all the wolves are pulled together. So we've taken out the signal of population structure. That's actually a noise here when we just want to look at hybrids. We care about whether animals are wolves, dogs, or hybrids. So that means we really only want to see these hybrid individuals, which you can see in red in the middle. However, I should also say that if you notice at the bottom of the image, you can see a cross as a marker for golden jackal. And up here, you can see that we cannot see jackals on this panel. They are just showing up among the other wild canids. So these reduced SNP panels, they allow us to identify wolves, dogs, hybrids, and the early generation backcrosses to parent taxa. And importantly, this was tested across Europe. So whether that wolf come from, or the hybrid comes from Finland or Italy or Portugal, doesn't matter. It really looks at the ancestry of the animal being wild or domestic. This also means, very importantly, that any suspect canids detected in non-invasive monitoring, where you have no actual animal, you just have a scat or a hare, it can be tested with a standardized method. And there are now various SNPs developed for non-invasive analysis, for example, also wildcat that are affected by hybridization, and a tool for Scandinavian brown bears, for example, where it's not issue about hybridization, but in using non-invasive samples for monitoring the brown bear population. If we look at pitfalls, we do, as I said, not see all species. We cannot see jackals on this initial panel. However, we did develop a second panel where we also added some jackal discriminant SNPs, again, after analysis on this 170,000 major canine panel. And it is important also to notice that detection of backcrosses is more difficult with time since hybridization. With these few markers, which are non-invasive, sample-friendly, we cannot detect reliably any third generation or older generation backcrosses. And we also need to caution that we tested this in Europe, so we are not sure if or how well it works outside the study area. For example, if you tested any samples from Asia, this is something that would need further study. So in conclusion, these new markers and tools, they promote faster data sharing among laboratories. And we can investigate any putative hybrids with shared, standardized, and importantly, also transparent published genetic methods. And we can improve our transboundary monitoring and therefore also conservation and evolutionary research at a scale that is relevant for many of these highly mobile species that we are interested in. So in final, just want to thank many organizations and thank you for inviting me and thank you for listening.